like you is to get this sort of impression of the diverse types of processes that we can now study and diverse types of interesting facts that we can harvest from the evolutionary history of life. And although there will be no solid laws, the regularities that we can find in the evolutionary process. Evolution is, of course, a stochastic process which is fundamentally unpredictable. Nevertheless, there are some soft rules, and this is what will this is what is worth noting. So, again, I promised not to talk to you about fossils, but instead of talking about the entire, or not to talk to you about fossils at length, but so I will not be focusing on the entire history on Earth, which is, as we discussed yesterday, pretty well documented, but there are individual instances in which the fossil record is so dense and so detailed that it is actually possible to treat it as a, as a detailed, almost as evolution observed directly. Perhaps the most spectacular of such instances over large time spans is the geology of the lake formations in the eastern United States, in the New Jersey, Pennsylvania, Virginia, and the neighboring states. So these lakes are formed in the course of tectonic activity. So that two large plates, the North American and African plates, have separated back in late Triassic, so that's about 230 million years ago, and they have been splitting away, forming the Atlantic Ocean, and as a result of that process, the rift lakes, so the rift valleys have been forming, which have been filling up by water in the middle between the glacial periods. And during the glacial periods, in the vicinity of glacial periods, those lakes would be filled up by ice and would dry out, and then they will expand some more, and the cycle will repeat itself again. Each such cycle lasted about 20,000 years, and each of these cycles gave rise to a layer of sediments a few meters deep. And so this whole thing continues for 30 million years. So there's actually a history of about 1,000 of such cycles traced in these sediments. And people have not cared to dig through the entire 1,000 of these cycles. Instead, they have studied one such cycle in detail, and basically you can trace the history of life in this lake, which has originated, survived for 20,000 years, and or like several thousand years, and died out right before your eyes. So there are these fish called Semionodos, and there are lots and lots of samples of these fish, and the sampling is so good that you can actually, for the different depths into this sediment, you can trace how many different species you can see. And you can see that we started with just a low number of species, and then a rapid process of speciation has happened, giving rise to lots and lots of species, say like 20 species, and then gradually as the lake was shrinking and drying out, these species were dying out. And this is very reminiscent of what is now happening in the African Rift Valley lakes, such as Lake Malawi or Lake Victoria, where we have the same process going on now and the same diversification process going on now. You have heard there are like tens, if not hundreds, of cichlid fish species that have originated there in the last several thousand years. So it's striking how much detail you can obtain from such a fossil record. So here's another instance. It's a much more recent evolution. It's about 20,000 years ago, and here people have been studying the morphology of shells and how it has changed in the course of evolution. And the problem there is that there are plenty of shells to study the evolution, but they survive in this sediment, which is very much mixed in the course of, which has been very well mixed for these thousands of years. So there is no way to use the sediment data, to use the geological layers to figure out how old an individual sample is. All these shells are mixed together. So what you instead need to do is you need to find some type of clock which resides within an individual shell itself. And fortunately, there is such a type of clock. You probably all know that the amino acids biogenically, all amino acids biogenically originate as L isomers, but throughout time, in the process of racemization, the L isomers turn into the D isomers, and until ultimately the 1:1 ratio is reached, which is sort of the equilibrium ratio. And so by the ratio of L, or let's see here, how does it work? Okay, so by the ratio of, the point is that by the ratio of one amino acid over the other, you can trace how recent the individual shell is. And so these are the most recent samples, and they have this peculiar, so sort of roundish morphology, but more ancient samples have this elongated shape. And you can see in detail how this shape has been evolving in time. Sometimes you can get data on entire communities, and this is the study field of Liz Hadley's lab in Stanford University, and what she does is she studies the middens, basically the trash piles of these North American rodents of the genus Myotoma. And these rodents basically pick up whatever they can harvest for food, plant residues, any type of mammalian food insect that they can find, and they breathe it all into their burrows, and the remains of those things stay there. So by the composition of the bones, so these burrows are then preserved for thousands of years. But so then you can come there a few thousand years later, and by the bone composition of those piles, you can actually trace the diversity and the composition and the ratios of different species abundances that have been there throughout the life course of this single trash pile. And so you can measure the abundances of different individual species, but you also can measure how the morphology of different bones, for example, has changed over this time course. Unfortunately, it's hard today to get DNA from those samples. I think Liz Hadley is trying to go there, but they just started. For marine and for freshwater sediments, you can get these sediment cores that you can extract from the sea or the lake bottom, that you can then study and trace, for example, the relative abundance of different spores or different pollen types in it, and again study the evolution of communities by that means. And finally, if you are lucky and you have genetic data, say for some fossil bone or some fossil remains that is of sufficiently sufficiently good quality, then you can trace the evolutionary history of that sample by studying its DNA, and this is something we'll again be talking in more detail later when we will talk about reconstruction of demographic history from DNA synthesis. And that's actually something you guys will be doing for us humans in one of the late computer labs in the course. Okay, so sometimes we can see two extant forms that are distinct but very similar to each other, and that covers sort of the entire space of intermediate forms. And in that case, it's almost as good as if we have seen the evolutionary process right in front of our eyes. So this is a species of potentilla, in Russian Lavchatka, in California, and it resides at very different heights, starting from the sea level and going up to 3,000 meters. And it is very well adapted to so it covers the whole slope in between, but it's very adapted to the particular localities at which it resides. So that if you do the transplant experiments and try, for example, to make the F1 hybrids of different individuals from different heights, how well those hybrids do, they do moderately well, and then they do better at different heights depending on what their morphology is more reminiscent of. So exactly what we would expect to see if we breed different ancestral derived forms with each other. So there's a number of such instances of ecological clients, that is, local ecological adaptations that together form some phenotypic continuum in our own species. The most notorious example is the diversity of skin color in contemporary humans, and here is the map showing the distribution of people with different skin colors globally, and you see that of course the darkest colored people mostly live in Central Africa, but people with dark color also reside in Australia and Oceania and in the New World, and just geographically you can immediately tell that these dark skin color here in the New World, for example, has originated secondarily, because we know that these Native Americans here have come across the Beringia down here. So this was a reversion to this dark colored state. And the genetics of that is now progressively better and better studied, and it's of course not a single gene, right? We can immediately tell that the skin color is not determined by a single gene. How do we know that?
With the gradient in skin color?
Yes, but even if we did not have that's true, even if we did not have this geographical data. So if in a marriage of a black and a white person, if it was a single gene, you would expect to see the 3-to-1 ratio of black and white offspring, right? If it were a single gene with a dominant, with one of the alleles dominant. We do not see anything like that. Instead, we see some distribution of skin colors which is intermediate, which immediately tells us that there are many genes involved. That's something we'll be talking about, in detail about when we talk about quantitative genetics. When lots of genes determine a single trait, we do not expect to see them in linear proportions. What we think now is that there are at least 10 major effect genes that determine the skin color, and probably lots and lots of genes with smaller effects, so with quite a large number of genes. And this is an instance of adaptation, apparently. The genes that are responsible for skin color show evidence for adaptation both in the low latitudes and in the high, in the low latitudes to the dark color, and lighter color, it is dark skin color is probably adaptive in the equatorial regions because it gives some protection against melanoma, against skin cancer. It is less obvious why the light skin color is advantageous in high latitudes, probably because it allows a better synthesis of vitamin D, although we're not sure. And there are a number of other instances in our own species in which we see selection that has operated on some alleles in some populations, but not in others. Here's one such notorious example. So this is a mutation, a similar mutation in the lactase regulatory sequence, which has originated in several independent populations about 10,000 years ago. It has similar mutations increasing the activity of lactase have arisen in the Western Europe, in the Crescent Peninsula, in the Fertile Crescent in the Middle East, in the Southeastern Asia, and in Africa. And these were mutations that made lactase expressed throughout the lifetime. And of course, the ancestral state for mammals is that the lactase, which is the enzyme that is used for processing milk, is only expressed in the childhood, before weaning, before the baby is taken off from the mother. Now, why would this mutation causing the expression, the lifelong expression of this gene, become advantageous about 10,000 years ago? Because of domestication of cattle, right? That's the most natural explanation. So apparently, if all other food is gone, but you have a cow producing some amount of milk, if you as an adult is able to consume this milk for your own good, that increases your survival. And so in time, we can date the spread of these adaptive mutations, and they do sort of coincide roughly with the origin of, with the domestication of cattle. And we do see the signs of selection in the genome at these loci. I have not told you yet how these figures work, but so that's something we'll discuss when we talk about tests for natural selection. But briefly, you have long identical haplotypes, long identical sequences, and low variability in the region around the advantageous mutation, because this advantageous mutation has spread fast through the population, and these DNA segments that have spread together with it have not had yet time to accumulate additional mutations. So as a result of such positive selection, the DNA segments become unexpectedly monomorphic and unexpectedly similar to each other. So that's a signal that one can use to identify past positive selection, one of several signals. And indeed, we see this strong signal of selection in this sequence. And that's not the only instance. There are a bunch of known cases of selection that has affected some human populations, giving rise to some local adaptations. Notorious examples include the adaptation to high altitude in the Asian peoples, some people from Nepal, for example, in Southern American people from Andes, lactose tolerance and pigment skin color we have talked about. There are lots of adaptations having to do with malaria resistance in Africa, where malaria is common. Many of those adaptations are apparently deleterious in the absence of malaria, so they do not spread outside of the region where malaria is endemic. Fine. Okay, so that's sort of cases where we can observe evolution not directly but by the trace it leaves on the current biodiversity. There are cases in which the evolution proceeds so rapidly that we can actually observe it in our lifetime or over the lifetime of several generations. And such rapid evolution is frequently associated with some types of conflicts. Most frequently, the conflicts between the host and its parasites. And the evolutionary theory tells us that a conflict between a host and a parasite should lead to evolution that is rapid and that is continuous. So it's something called the Red Queen scenario, in which the host continually evolves defenses against the parasite or the pathogen attacking it, while this pathogen continuously develops ways to circumvent these defenses. So this is an arms race which is continuous and which can, in theory, go on indefinitely until one of the species goes extinct. And indeed, many of such instances of rapid evolution are attributable to this host-parasite coevolution. So these are Daphnia invertebrates found in a lake in Belgium, among other places, many other places. And so they lay eggs, and these eggs are deposited in shallow sediments. And then if you can extract those eggs, date them, and hatch them. And from the same date, from the same year, you can actually extract the parasites that would be attacking this Daphnia. So what people did was they extracted the parasites and the Daphnia from the corresponding layers, and they found that the parasites were more efficient in attacking the Daphnia from this layer than the Daphnia from the slightly later or slightly earlier layer, which is exactly what we sort of expect. So the parasite was perfectly adapted to the Daphnia of the current year rather than those of other years. Another instance of host-parasite coevolution comes from a notorious parasite, insect parasite called Wolbachia. So Wolbachia is a bacteria which is intracellular, which lives within cells of an insect, and it is only transmitted vertically from mother to daughter. It is not transmitted to sons. So as long as it is unable to be transmitted to sons, it doesn't make sense for it to allow sons at all. So what it does is it changes the sex ratio of the offspring of insects, making it very much female-biased. When the Wolbachia first infects an insect species, an insect population, usually the sex ratio becomes almost 100% females. However, there is very strong selection, of course, in the insect population to restore the sex ratio to the original one, because you cannot do without males at all. So theory tells us that the 1-to-1 sex ratio is very strongly selected for. And here people have studied the sex ratios in several island populations of these butterflies that have recently been infected by Wolbachia, and immediately after infection the sex ratios were very biased, but rather rapidly they have been restored back to the normal 1-to-1 ratio due to the evolution of these instances. So again, an instance of very rapid evolution over a time scale of years in a host parasite system. Finally, our own species provides a lot of very serious opportunities for novel evolution, and actually triggers evolution in many species because we disturb the environment so much. Humans are the most serious invasive species on Earth. It gets into some kind of contact with the enormous diversity of different species of very different kinds. And by doing so, it triggers their evolution. And here's a notorious example that many of you are probably familiar with. That is a butterfly called peppered moth, Biston betularia, in Britain. And there are two photos. And how many butterflies can you see in those two photos? 4? Okay, the people with good eyesight can see 4 butterflies, right? So this is a birch covered with lichen, and there is a black butterfly here, but there's also the white butterfly. I'm now showing the outline of this butterfly here, okay? And there's a birch without the lichen, and there is the white one here, which is much more obvious, and the black one, which is nearly invisible. So if it's invisible to us, it will be also invisible to the predator, and there are experiments showing that, and you can easily see that the optimal phenotype will very much depend on the environment. And the environment has been changing. The lichens covering the birches have been dying out due to the air pollution, mainly by coal particles in the late of the 19th century and in the early 20th century. And as a result, the black melanistic morph has spread throughout the Britain. Now in 1950, the Clean Air Act has been approved in the United Kingdom, and the coal pollution at this scale has been prohibited, and as a result, the lichens have started to spread rapidly, and the white typica morph has also started spreading very rapidly. You can actually trace this process. So the crossed area is the area of high density, over 90% density of the melanistic morph, and you see that it is contracting rapidly because the typical form spreads rapidly back to the pre-pollution situation. And people have used this data to actually estimate the strength of selection in favor of the white genotype. And it turns out, white phenotype, and it turns out that this selection is pretty substantial and rather strong. When we talk about the coefficients of selection, we'll see that this is strong selection, which is pretty unusual, except in such radical cases in which something very, some very major environmental shift happens. Recently, in our lab, Nadia Tsirikhanova has also studied the selection in natural environments in small stickleback fish that are adapting to the fresh water, moving there from sea, and this is similar to the selection coefficients she has observed there as well. Fine. So, what else do humans do? Well, humans mine for metals, such as zinc, copper, and so forth, and in doing so, they produce these huge piles of rocks with high metal content, and the local plants have to adapt to this high metal content rapidly, and this is one such instance. The rock crest Arabidopsis halleri has actually produced populations tolerant to high content of metal throughout its habitat, and these populations are not non-sensitive, are resistant to the high metal content of these mining grounds. Perhaps surprisingly, if you build the evolutionary tree, the phylogenetic tree, and onto this tree you include both the metallified and the non-metallified populations. So MM is the population which was ancestral, and the M population is the population that has evolved to be resistant to these deposits. You see that usually what happens is the M populations are closely related to different ant populations are closely related to some non-metallified populations, as opposed to them all forming one large plate on this tree, which means that this metallified phenotype, this metal tolerance, has been evolving independently several times. Okay, so it arises locally due to some local adaptation, not as a result of one single ancestral mutation, and then this phenotype spreading throughout this state in the United States. So people harvest species, and species evolve in response to us catching them and eating them. And it probably happens on the largest scale for the wild populations. It happens on the largest scale for fish. And by fishing, people impose selection that is not there without fishing, in the absence of fishing. In the absence of fishing, the fish are selected for large size because they are, in that case, less vulnerable to predators, for example. And if they're predators themselves, they're more efficient as predators. Conversely, when humans fish, what they frequently do is to pick out the largest individuals, keeping the small ones intact. And if you do that on an industrial scale, you just usually have these nets with a large mesh size, and the small fish slides through that mesh, but the large fish stays in that mesh. Okay? And as a result of that, in the seas globally, the fish have evolved to become smaller. So there has been selection by our own species, which counteracts the selection that is naturally imposed in this marine environment. The fish has dropped in their age at which they become mature, so they now start breeding at younger age. They grow more slowly, in addition to their lower survival. And, you know, here one could argue that the ecological thinking and the evolutionary thinking proposes the different solutions. Because if you go fishing in the United States, for example, you just take a rod and go fishing, and you get a license. And what people normally do, if they catch a large fish, then they can take it with them. If they catch a small fish of the same species, they're supposed to release it back into the pond, because the idea is that ecologically you should allow the small fish to survive, to grow and to breed and to produce lots of offspring on its own, while the large one will sort of die soon anyway. That makes sense ecologically, but that does not make any sense evolutionarily, right? Because evolutionarily what happens is you select for the smaller size, which is not what you want to do. And so we have affected many different populations in this way, such as cod, and this is an ongoing large, major ongoing problem. So this is a nice example that I think is illustrative. So there are these pigeons in towns, which we all are very familiar with, and the ancestral coloring is this gray coloring with two black stripes. Now, for some reason, when the pigeons move into the large towns and cities, they evolve this melanistic dark coloring. And there have been, we don't really know why, there have been some work suggesting that that's associated with that trait is somehow linked with aggressiveness, but we still do not know whether that's a pleiotropic effect, so the species, the pigeons actually evolved to be aggressive in the cities, or these two traits just happen to be next to each other on the chromosome, and are linked genetically but not functionally. Nevertheless, what happened during the World War II was that in large cities, of course, many of the pigeons got eaten by the starving population in the former Soviet Union. And then, after the end of the war, new pigeons had to move into the cities from the rural villages outside the cities. And when they moved out, they were sort of gray with black stripes, but then this melanistic phenotype has again evolved rapidly, which means that the selection for it, whatever it is pretty strong. People don't really know what it is. That's a fascinating example in which the rapid adaptive evolution has been caused by introduction of a species into a new environment. And that's one of the many cases in which such rapid adaptation has happened in Australia, because Australia has not hosted much of the fauna that we humans were then able to bring there. So people at the northeast of Australia were sort of suffering from the mosquitoes and from small insects at the sugar cane plantations, and someone had this brilliant idea, why don't we bring a frog from the America to fight with these mosquitoes. So these are the cane toads ufomarinos, which used to be about this large, so when they were released, they're introduced and released there in the Australia, what happened were two things. First, they started growing very rapidly in size, and this is the size they have some individuals reach now. Second, they started expanding very rapidly. And the striking thing is that their area is still expanding, but it is expanding with acceleration. So every next year, the front moves forward more than it did the previous year, which sort of means that these things evolve to move faster. And that makes total sense, right? If you're living here and behind your back there is a swamp filled with other toads like yourself, but in front of you there's whole of Australia, which is still vacant and has all the insects you might want in it, it totally makes sense to move fast. And that's exactly what happens. And so the Aboriginals and the Australians are looking at these toads now and, scratching their heads and thinking all the nice words about this brilliant guy who had this brilliant idea to bring them in the first place. So that's again a rapid adaptation caused, triggered by the human activity, in this case introducing a new species into a non-native environment. So here is another such case, which is something that totally could not have been predicted. There is this thing called, it's a little bit complicated, so stay with me for a couple of minutes. So there's this thing called P element, which is actually a mobile element. It's a selfish DNA which transmits itself between the different genomic regions in insects. And originally it has been present in the New World species of Drosophila. So Drosophila is, first of all, genus Drosophila, the fruit flies, is a very is a globally distributed species, but there are different. It's a globally distributed genus, but there are different species in different parts of the world. So there are New World species historically there were Old World species, and the familiar Drosophila melanogaster, the most broadly used lab species, is one originating from the Old World, from Africa. Now, originally this P element was limited to the New World species, and what this P element does is that if just one of the parents in a cross has it, then all the offspring have it. So as a result, it spreads very rapidly through the population. And all the New World Drosophilas are very highly infected with it. However, melanogaster was free from it. But in the early 20th century, actually, way before that, in the 18th, 19th century, Drosophila melanogaster has been brought into the United States, either with the African fruits or with the slaves. We do not really know, and has spread throughout the United States. And initially, in the early 20th century, the Drosophila melanogaster did not have any P element in the United States or elsewhere. However, at some point of time, this P element has been transferred to it from another Drosophila species, probably from Drosophila willistoni, from from New World species, probably through a mite which first beat Drosophila willistoni and then Drosophila melanogaster, and then it has started to spread throughout this population of melanogaster, not just in the New World, but also in the Old World, also back in the Eurasia. And so there was this wave of P element moving through the population that people have sort of been observing because it disrupts the genetics of Drosophila to a certain extent. And about 50 years ago, probably 40 years ago, Sergei Nuhdin, who is now at the University of California at Davis, then a geneticist at the Moscow State University, has tried to find Drosophila melanogaster that were still P negative, and to try to do that, he packed a backpack and went to the region that he thought was most remote from the New World on the globe. So he went to some, I believe, Tajik mountains up high to the Kishlaks there, and picked up several some Drosophilas from there and brought them back to Moscow and studied them, and it turned out that they all already had the P element back then, or by then. So this P element has spread like a wildfire throughout the population of Drosophila melanogaster globally in just several tens of years, just several decades. So that's really fascinating how efficient and strong and selection favoring and invasive species, or in this case an invasive transposable element, can be. A century ago, just think about that, a century ago, some mite has beat some Drosophila melanogaster somewhere in the United States, and then a century later, all the Drosophila melanogaster globally, in Tajik villages included, they are all have this P element which is spread throughout those populations. I think it's pretty fascinating. Okay, let's make just a 5-minute break, okay, and literally 5 minutes.